Hi, my name is Trinity. I am a graduating senior majoring in cellular and molecular biology from New Jersey. And the article that I have chosen is the comparison of 3 molecular assays for the detection and molecular characterization of circulating tumor cells in breast cancer. So the objective of this study was to compare 3 different molecular assays to detect and determine the molecular characterization of circulating tumor cells in primary and metastatic breast cancer. So the molecular assays that were used were the RT-qPCR, the multiplex RT-qPCR, and the ADNA-test breast cancer. So circulating tumor cells are an early marker of response to systemic therapy. It gives information on relapse and progression of a disease. The advantages are is detects viable circulating tumor cells, and it's an early response to therapy based on the molecular characterization of circulating tumor cells. They are also personalized for target treatment. Circulating tumor cells have a low frequency, so the methods that were used needed to be of high sensitivity and specificity. The expression of different genes can increase circulating tumor cells positivity in breast cancer patients, which can provide information about clinical management. The multiplex RT-qPCR targets different mRNA transcripts, and it uses the genes CK19, HER2, MAGE-A3, and PBGD, which was the control. The ADNA-test breast cancer isolates CTC by immunomagnetic beads with antibodies against MUC1 and an epithelial cell adhesion molecule EpCAM. Multiplex RT-qPCR happens after, and then detects GA7332, MUC1, and HER2. Beta-actin is the control that it also detects. The RT-qPCR detects CTC through CK19 expression. So the materials would be obviously patients for the study. So 254 patients were enrolled in the study for early breast cancer, 51 patients for verified breast cancer or metastatic breast cancer, and 30 healthy female blood donors were used as a control. The ADNA-test breast cancer kit, the Dynabeads mRNA direct microkit isolates mRNA from lice cells. The SensaScript reverse transcriptase uses reverse transcription because of the high sensitivity, and then the multiplex RT-qPCR. So onto methods. So the circulating tumor cells are first isolated. So blood samples are isolated and analyzed for circulating tumor cells using the ADNA-test breast cancer kit. And that kit enables immunomagnetic enrichment of tumor cells via epithelial and tumor-associated antigens. The blood samples are isolated and stored for 48 hours, then incubated with an antibody mixture. The cells are then extracted by a magnetic particle. So the RNA synthesis is next from the circulating tumor cells and cDNA synthesis. So the mRNA is isolated from lice cells using the Dynabeads direct microkit. The SensaScript reverse transcriptase is used for high sensitivity, and the cDNA quality is compared with the control genes of beta-actin and PBGD, and 305 out of 541 were enrolled in the study because they were positive for both controls. CK19 RT-qPCR is a singleplex RT-qPCR which detects the expression of CK19. Multiplex RT-qPCR detects the expression of CK19, HER2, MAGE-A3, and PBGD, which is the control. And the ADNA-test breast cancer detects HER2, MUC1, GA7332, and all cDNAs and beta-actin as the control. So for the results, for the CK19 expression of RT-qPCR, 14.2 early breast cancer patients detected CTC, 41.2% of the verified metastatic patients detected CTC, and 3.3 of the healthy individuals detected CTC. For the multiplex RT-qPCR, it was considered successful if at least one tumor-associated transcript was shown in the PCR fragment, so CK19, MAGE-A3, or HER2. So for the early breast cancer patients, it was 22.8% showed expression levels of circulating tumor cells, 9.8% showed expression levels of CK19, 4.7% showed expression levels of MAGE-A3, and 11% showed expression levels of HER2. For the verified metastatic patients, 39.2% showed expressions of CTC, 35.3% showed expression of CK19, 7.8% showed expression of MAGE-A3, and 15.7% showed expression of HER2. In the healthy individuals, there were 6.6% that were positive for CK19, HER2, MAGE-A3, and PBGD, which is the control. The ADNA-test breast cancer was considered successful if at least one transcript detected was at a concentration of 0.15 nanograms per microliter. So in early breast cancer patients, 16.5% showed circulating tumor cells, 3.7% showed GA7332, 5.5% showed MUC1, and 10.2% showed HER2. In the verified metastatic patients, 54.9% showed circulating tumor cells, 39.2% showed GA7332, 45.1% showed MUC1, and 35.3% showed HER2. In the table 2, it shows how CDC was detected in breast cancer cells from RT-qPCR, multiplex RT-qPCR, and the ADNA-test breast cancer, and it compares all 3 molecular assays in that table. Table 3 compares the molecular characterization of the circulating tumor cells with each molecular assay that was used. So the multiplex RT-qPCR, the singleplex RT-qPCR, and the ADNA-test. Table 4 shows the comparison of the CK19 detection between the RT-qPCR assay and the multiplex RT-qPCR assay. So in this one, the ADNA-test breast cancer kit was not used. Figure 1 shows the circulating tumor cells molecular characterization in early breast cancer cell patients, and figure 2 shows the circulating tumor cells molecular characterization in verified metastatic patients. So for the conclusion, quality control is obviously important in detecting circulating tumor cells, analyzing it, and the characterization of circulating tumor cells. It has an effect on different molecular transcriptions on detection of circulating tumor cells. So the same target detected in the same cDNAs with the same set of primers and probes gives a good concordance between RT-qPCR and multiplex RT-qPCR, but there was no correlation between using a different set of primers, but with the same DNAs. CTC positive with different transcripts causes discrepancies, which is circulating tumor cells would be low in breast cancer, in early breast cancer patients, but high in verified metastatic patients, which that also shows comparable results even while targeting other transcripts. And the CTC heterogeneity is very important for detection by different molecular assays. Thank you.